come up and grab a whiteboard and grab an eraser and just do, if you want to do it by yourself, you're welcome to. If you want to just get in like a group of 2 or a group of 3 sometimes, bouncing ideas off of each other really does help a lot. Here's a whiteboard, here's an eraser, here's a marker, and go through these. Try not to look on the board. See if you can go do a DNA double strand. Just make up a DNA double strand. Make an RNA single strand. I'll pull up the codon chart so then you can read the codon chart and make your own amino acid sequence. What is DNA? What is RNA? What is an amino acid? Go through those together really quickly. And then I will go through kind of what I wrote on the board because I'm not going to talk about that during class. We're done with that section. So yeah, if you guys want to get together and discuss those things, totally up to you. Oh, enzymes also. The enzymes for every step. That we're talking about the structure of the ribose molecule. So that's why there is a directionality to it, because you can only attach to a certain point on the molecule. So that's why we care about 5'3' is because you can attach it in one spot. The DNA polymerase can only attach in one spot, and the RNA polymerase can only attach in one spot. So just as long as you know what the molecule looks like in the prime, what the importance of the primes are. So remember, this is 1, 2, 3, 4, 5. So 5 prime tells you that's that direction, 3 prime tells you it's that direction. So if you had a double-stranded DNA, which is both of these red, that's our double-stranded DNA. The helicase came and broke all the hydrogen bonds. The topoisomerase untwisted. You have the single-stranded binding proteins to help you. Now the DNA polymerase 3 is going to come in and add. So what Dr. Myers had a good point is, this is how I always remembered it. Read, synthesize. So 3, 2, 5, 5 to 3 is how I always remembered it, because R comes before S in the alphabet, and 3 becomes before 5 in the number line. So that's how I always remembered it. But Dr. Myers had a good point, is that you don't really even have to know the synthesize. If you just know 3 becomes before 5 on the number line, and you just know that that's the way the polymerases read. So when you're talking about reading, is the polymerases are always going to read this direction. So they're going to add here according as they read the primary strand.
This is DNA and polymerase 3.
Yep, this is DNA pol 3. Mm-hmm.
And then DNA and polymerase 1 reads from 5 to 3.
DNA polymerase 1 just comes in and chops. And yeah. From 5 to 3.
Because it goes the opposite direction.
Yeah, I didn't know that. Is that a thing? That DNA pol 1 goes in a different direction than pol 3?
Well, because if you think about it, like if one's going this way from 3 to 5, and the other one's putting in this way.
I don't think so, but let me Google it. DNA pol 1 reads in a different direction than pol 3. Let's see.
No, I think potentially the confusion is like, and on the vocab, it says that it reads 5, it allows it to read 5 to 3, but it's still reading 3 to 5, it's just in that.
Oh yeah, so DNA pol 1 can do either, according to PubMed. So DNA pol 1 is like the exciser. But I think, correct me if I'm wrong, but what I think that you're saying is there's naked spots. And who fills the naked spots? That's what you're asking. Okay.
Yeah, and it's
so,
going the other direction.
no, they go the same direction.
Okay.
That's the, yeah, that they are going the same direction. So if we had DNA, sorry, do you have a way way to clarify it?
So polymerase 1 it is like proofreading too, right?
DNA pol 1 is the proofreader.
So I think when it goes back, it's just a proofread, right? It doesn't actually.
Yeah, it doesn't really like synthesize directionality. It comes in and cuts
Right
and
behind
adds,
it,
cuts and
right?
adds, yeah. Okay, so let's say, so without thinking about 5' to 3', then you have this, your second strand down here. DNA polymerase 3 is gonna come in and add, so say it starts right here. This is the ORI. Then they have to always go in the same direction. So on this strand, they're gonna, I guess we should add, so we should go 5. 3. So this one has to go this way, because it's reading from 3 to 5, right? Is everybody with me on that one? It's reading from 3 to 5. So it's reading this strand and adding nucleotides to match it all the way along. So this one, all polymerases have to move, all DNA polymerases and RNA polymerase have to move in the same direction. So this is going to be reading also from 5 to 3.
You mean 3 to 5?
3 to 5.
That was
From
right.
3 to 5. Oh yeah, that was right. From
Yeah.
3 to 5. Okay, so here. So now there's naked pieces behind it, and there's naked pieces behind it. Well, what happens is a DNA polymerase is just gonna come up behind it and make copies. So let me see if I can find a good, well, you know what, I think I just have to post a video, because what you're not seeing is that the more it untwists, the more there's naked pieces.
Right.
And so when there's naked pieces, it'll come and jump in here and keep going, but then there's more naked pieces. So then it'll come up here and keep going. More naked pieces come up and keep going. So those are the Okazaki fragments
Right.
over here, and there's the Okazaki fragments over here, but the DNA polymerase always goes in the same direction.
So it's still gonna add it on 3 to 5, it's just opening more fragments, or opening more
Unread.
unread ones.
Yes.
And
So
that's
it's-
always on the lagging.
It's, yes, but I don't really love that, because this is lagging and this is lagging.
Right.
So there's gonna be a lagging piece.
But like the fragments
and
are
a
always
leading
on the
piece.
lagging section.
The fragments are always on the lagging section, or always on the section that has to jump to catch up and jump to catch up. This one can move all the way around the circle till the opposite side. This one can move all the way around the circle to the opposite side. But there's gonna be a whole bunch of naked DNA over here, and it has to come and catch up and come and catch up as it untwists, and then ligase comes and ligates those little Okazaki fragments.
The videos are really good.
The videos are good.
Yeah.
If you find a good video, will you send it to me and I'll post it? Okay.
So is it DNA polymerase 3 that's doing it on both sides?
All DNA polymerase 3.
And then the DNA polymerase 1, sorry I keep saying it weird,
Yeah.
is the one that just proofreads it,
Yep.
make sure it's correct.
Mhm.
And then the Okazaki fragments that are in between the 3,
Mhm.
right, but then the ligase comes and fixes the Okazaki fragments.
Yep.
Okay.
Or Okazaki fragments only on the lagging strand.
Only on the lagging strand here and the lagging strand here. Maybe it'd be better to call it a lagging pieces. Because they're the same strand. It's just there's a lagging section, and here's a lagging section.
Okay.
Yeah.
So, and it's 3 to 5, or
So it's still gonna come in and move this direction. And then it'll unwind more, so there's gonna be, so if the Pac-Man's moving this way, as it unwinds, there's gonna be more naked stuff this way. So it's gotta come in, another Pac-Man's gotta come in. Unwind more, another Pac-Man has to come in. So these are the fragments. This one can just go all the way around the circle. But this one's gonna run into this one. This one's gonna run into this one. This one's gonna run into this one. So those are the fragments that have to be ligated.
okay.
Yeah?
Okay, yeah.
Okay. Okay, well, I'll post a good video of it. If you guys have a good video, if you can send it to me, but also I'll do some research on the internet and try and find you guys some good videos. Also, Dr. Rosier and Dr. Welsh can definitely help you with that for sure. So make sure that you know all the enzymes, DNA pol 1, DNA pol 3. Here, this is DNA polymerase 3. This is RNA polymerase that polymerizes RNA. Here we have a ribosome that's drawn in a really funky way. Let me get my colors back. Sorry, I'm taking up your guys' whole desk. Thank you. Okay, so here is, so tell me really quick, this is the anticodon that we don't really care about, but let's just do it because it'll match. So U matches with what? A. C matches with? G matches with? So we're not reading this anticodon, we're reading the codon. So GCU matches with what amino acid? So here's
Alanine.
the first, yep, G. Here's the second base, C, U, alanine. So that's ALA. Then here, just our anticodon, we don't really care about, UA. Look at this one, UAG. UAG matches with what?
Stop.
Stop. So actually, when this one isn't attached to any amino acid. So we have one amino acid that's part of this strand. If we went on and on, it would attach the amino acids as it goes. When it runs into a stop codon, it falls off, and you have the amino acid chain, which is also called a polypeptide, which means protein. And so proteins, those are all proteins. Every enzyme that breaks up glucose, that goes through all those chains of glycolysis, all of the cytochromes, all of everything in our body basically is run or governed by proteins in some way. So it forms the proteins that run our whole body. Good? Okay, well, let's move on. So we have a lot to get through, and I want to make sure that you guys understand this section. So please, 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 let's do some Zoom calls this weekend. Talk to Dr. Welch, talk to Dr. Rosebeer. We can get together, whatever you want to do, because we're moving right along into gene regulation. Everybody ready to go? Feeling good? Yeah? Okay. Okay, so we talked about